Historical background and general technique of plant tissue culture. Plant tissue culture is the cultivation of plant cells, tissue, or organs on a suitable nutrient medium under controlled conditions aseptically. It is carried out in vitro under sterile conditions, and the plant parts which can be used are either leaf, stem, roots, or or cells or tissues or organs can be used. And the plant tissue culture is possible because of the inherent capacity of the plant cell, that is totipotency. The plant cell has a capability of producing a whole plant from a cell. So the question arises, why plant tissue culture? Earlier there is conventional breeding, which is now also it is being in use, and it is widely used for crop improvement, but the only limitation it has is the time and the scope. It takes around 6 to 7 generations for the production of the pure lines, but in case of plant tissue culture this time can be saved. Moreover, haploid cultures can be produced and the genetic transformation can be done easily. Historical development of plant tissue culture. In 1902, Herbert Land proposed the concept of totipotency. In 1904, Hanning successfully cultured a mature embryo. In 1925, Lebach used embryo culture to rescue hybrid progeny from interspecific crosses in Linum. In 1930, discovery of indole acetic acid and role of vitamin B was crucial for successful callus culture, and growth of tissue in vitro was established. In 1939, first continuously growing callus culture from cambium tissue was established by Guthrie White and Nobicot. In 1944, Skoog reported shoot and root differentiation from tobacco pith tissue culture in vitro. In 1955, discovery of another plant hormone, that is kinetin, enable initiation of callus culture from differentiated tissues. In 1957, Skoog and Miller demonstrated that root and shoot differentiation is regulated by auxin cytokinin ratio. When the auxin to cytokinin ratio is high, root formation is there in the callus. When cytokinin to auxin ratio is high, then there is shoot formation. When these are in nearly equal, then the callus proliferation is there and there is nearly no organ differentiation. From 1958 to 59, uh, Reinvert and Stewart independently reported somatic embryogenesis from carrot tissues. In 1959, Braun regenerated the first whole plant from a mature plant cell. In 1960, Cocking achieved protoplast regeneration using cell wall degrading enzyme, advancing protoplast culture. In 1964, Indian scientists Guha and Maheshwari produced haploid plants from pollen grains by anther culture in Datura, initiating polar culture technique. In 1972, Carlson and co-workers produced the first somatic hybrid plant by protoplast fusion. In 1950s to 60, major contribution by Gottfried and Pierre White led to the development of modern tissue culture media. Culture room should have controlled temperature. It should there should be light, and the automatic timer for photoperiod control should be there. Shaker for liquid or suspension culture should be there. Power backup should be there to prevent loss of experiments during power failure. And talking about the culture vessels, they can be glass or made up of plastic. Glass vessels are generally cheap, they are reusable and autoclavable. These are made up of either borosilicate or pyrex. Uh, Plastic vessels are disposable and autoclavable, but in the long run, these are expensive. Common culture vessels used are culture tubes, flasks, petri plates, wide mouth bottles, cotton plugs, and polypropylene metal caps used for the closure. Coming to the washing procedure, first soak the vessels in the detergent, then wash them with the brush, then rinse with the tap water, then rinse with the distilled water, and dry at 75 to 80 degrees Celsius. In case of the contaminated vessels, it is recommended to autoclave them first and store these vessels in a dust-proof cupboard. There are various methods for the sterilization of media, vessel, instrument, and plant material. Uh, the sterilization is done to eliminate the microbes. The various methods are dry heat, flame sterilization, autoclaving, filter sterilization, 70% ethanol wiping, and surface sterilization. Dry heat sterilization, it is usually done at 160 to 180 degrees Celsius for 3 hours, and it is used for empty glasswares or Teflon plasticwares. Glass bead sterilizer, it is it the sterilization is done at 300 degrees Celsius, and it is commonly used for tools. Flame sterilization, instruments dipped in 95% of alcohol and flamed. Mouth of the culture vessel flamed before inoculation. Moreover, the inoculating loop are also flame sterilized. Filter sterilization. It is used for heat labile substances like hormones. It uses membrane filters which are generally less than 0.45 micrometers. Autoclaving. It is in autoclaving the temperature is generally 121 degrees Celsius at 15 psi pressure. The time is generally 15 minutes for small volume media and 40 minutes for large volume media. It is used for media, culture vessels, contaminated cultures, and autoclave opened only after zero pressure. Should be open only after zero pressure. Laminar flow cabinet. It provides aseptic working condition. It uses HEPA filters which removes particles more than 0.3 micrometers. The air speed is generally 1.8 kilometers per hour. Nutrient media, mostly synthetic or chemically defined media, is used for plant tissue culture. The composition is constant and reproducible. Different media developed for specific plant species and different culture purposes. The nutrient media contains carbon source. Since culture tissue are often photosynthetically inactive, an external carbon source is essential. Generally, sucrose is the most common carbon source. Other sugars like glucose, fructose, or maltose can also be used. Functions include it provides energy, it supplies carbon skeleton for biosynthesis, it maintains osmotic balance of the medium. Inorganic nutrients. There are two types of nutrients, macronutrients and micronutrients needed to the plants. In macronutrients, these are needed in relatively higher concentration. These include nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur, while micronutrients are required in smaller concentration. These includes iron, manganese, zinc, copper, boron, molybdenum, cobalt, and iodine. Growth regulators are also very important for the growth of the plants. Growth regulators control cell division, elongation, differentiation, and organogenesis. These are used in very small concentration. Auxin is a growth regulator, but a plant hormone. It includes indole acetic acid, IBA, NAA2,4-D. It is helpful in cell elongation, callus induction, and root initiation. The second hormone is cytokinin, also known as kinetin, BAP, and zeatin. Functions include it helps in cell division, shoot initiation, and delay the senescence. Third hormone is gibberellin. It promotes stem elongation and embryo growth. The another hormone is abscisic acid. It inhibits growth, promote dormancy, and embryo maturation. Complex organic additives. These supplements enhance growth when the exact nutritional requirement of the tissue is unknown. This includes vitamins, amino acids, and natural extractives. Its functions include it promotes cell growth and differentiation, improve regeneration efficiency. Then selection of a suitable media. Selection of a nutrient media depends on the plant species and genotype, type of explant, objective of culture, growth regulator requirement. Commonly used media for plant tissue culture are MS media. It is it is widely used. It has high salt concentration suitable for many plant species. White's medium, it is used for root cultures. And Gamberg B5 medium, it is suitable for suspension culture and legumes. Thank you.